Hello friends, welcome to my channel. Today let's talk about the molecule Ceralasertib. This molecule was developed by AstraZeneca and currently under clinical trial phase 3 stage for the treatment of advanced solid tumors. Here is the chemical structure for the molecule. Now I will be discussing about the synthetic routes. First I will discuss about quickly go through the initial medicinal chemistry synthetic route, which includes this chiral morpholin derivative as the starting material, which is commercially available as well as can be synthesized from D-alaninol in 3 steps. So reaction includes base mediated SN-AR reaction with this dichloropyrimidine, followed by lithium borohydride mediated reduction of this carboxylic methyl ester to this primary alcohol and conversion of this alcohol into mesylate by using mesyl chloride, followed by nucleophilic displacement with the help of sodium thiomethoxide to get this sulfide, and enzymatic asymmetric oxidation of this sulfide to sulfoxide and conversion of the sulfoxide to this sulfoxamine with the help of hypervalent iodine reagents catalyzed by rhodium acetate. And next step is deprotection of this trifluoroacetyl group, and followed by cyclopropylation with the help of bromochloroethane molecule to get the intermediate this 11, which was converted into the corresponding HCl salt, and the final step was the palladium catalyzed Suzuki coupling reaction with this boronic ester. Now the overall process includes some key challenges, like in terms of economical advantage. If you consider, there are some challenge like this chiral morpholin derivative is quite expensive raw material, so it is better to introduce this molecule at the later stage of the synthetic sequence. And also this rhodium acetate is also quite expensive catalyst, so use of that one in commercial scale is also a big challenge. So, and here in the cyclopropylation step, there are some unwanted byproduct through the over alkylation on the nitrogen center. So to address all these challenges, they developed the overall process during the scale up. So here is the scale up route. This is the overall scheme. Now I will be discussing each of the steps in details. So here they started with this 2,4-dibromobutyrate molecule and treated with the sodium thiomethoxide in solvent 2-methyl THF and methanol as co-solvent. And this reaction selectively happens through C2 position of this ester. So here the use of co-solvent methanol actually help to improve the solubility of the sodium thiomethoxide and it enhanced the reaction rate. Now, treatment of base like sodium hydroxide helped the cyclization to lead the cyclopropyl ring formation, and later the use of excess quantity of aqueous NaOH solution facilitated the saponification reaction, means this methyl ester to carboxylic acid. Now, as they have used 2-methyl THF as the solvent, no workup was required, only they did the layer separation, phase separation, and telescoped this intermediate 19 for the next reaction. So next conversion was to convert this carboxylic acid into the beta keto ester. For that one, they treated this carboxylic acid with this CDI to form, CDI is carbodiimidazole, to convert this one into imidazolide. That reaction also in 2-methyl THF, it worked very well. Then they introduced this potassium salt of this active methylene compound, and it react with this imidazolide, followed by decarboxylation under mild condition in presence of triethylamine and magnesium chloride, helped the formation of this beta keto ester intermediate 20. Now, to convert this intermediate into this pyrimidone, they first reacted with ether molecule in presence, urea in presence of base, but they got this pyrimidone with very less yield. So they switched to this thiourea, and in that case, it worked very well to get this thiopyrimidone. With very good yield it was obtained. And here you can see they have done some base selection optimization. In all other places you can see a lot of other byproducts were forming, but when they used this potassium tertiary butoxide, they could isolate this desired product, very good yield, like 85%. And now this conversion of this thiopyrimidone to pyrimidone was done using chloroacetic acid in water under heating condition. This one is very good reaction. They obtained this intermediate 31 in very big scale, like more than 100 kg scale. Now they converted this pyrimidone into the dichloropyrimidine using phosphoryl chloride in presence of diethyl phenylamine under heating condition. Now, after the reaction, the quenching process was very crucial at this stage. Like when they quenched that reaction mixture using sodium hydroxide solution and keeping the pH approximately 14, like under highly basic condition, then partially hydrolyzed chloropyridone was formed along with some reversal of the product into the starting material. Only the desired product was possible to isolate when they have used aqueous sodium acetate solution along with simultaneous addition of sodium hydroxide solution, and they kept pH in between 6 to 7, and they got this desired intermediate 32. And now they introduced this chiral methyl substituted morpholine This is one base mediated SNAR reaction. Here also the key challenge was regioselectivity, because it contained two chloride group. You can see selectively they are required at this position, but you can see they could not make completely regioselective product. The best condition they used in presence of triethylamine and potassium carbonate. That time they got the mixture of two isomers. The desired one was 83% and undesired one 17%. Now they did one good thing, like they separated these two isomers in very good way, like they did the phase separation MTB and water, and when they maintained uh, pH of that solution using 4 molar HCl, that time they have seen that desired isomer completely coming under aqueous solution and the undesired isomer is going into organic layer. In that way they have separated the desired isomer one. And the next step they performed the enzymatic chiral oxidation of sulfide to sulfoxide. So they have used this BVMO, that is the Bayer Vinegar monooxygenase, and they have used the NADP, that is nicotinamide adenine dinucleotide phosphate as the cofactor, which is getting oxidized to NADP plus during that reaction, and to recycle that one they have used this CRED, that is ketoreductase enzyme, and which is oxidizing that isopropanol into the acetone. So in this way they are getting this chiral molecule intermediate, very good yield as well as very highly enantiomeric purity, like more than 99%. Now to convert this ketoxime, this sulfoxide into that sulfoxamine, they performed this hypervalent iodine reagent mediated oxidation. They have used phenyl iodine diacetate as the oxidizing agent and ammonium carbamate as the nitrogen source. They have used the toluene as the solvent and methanol as the co-solvent. And after isolating that product, they have treated with HCl in IPA to get the HCl salt of this molecule. And then they introduced this boronic ester. That boronic ester was synthesized by from this bromo derivative of this heterocyclic molecule in presence of palladium catalyst, and B2P2 was used as the boron source. Isopropyl acetate was used as the solvent. Here the byproduct was like this dehalogenated product, but they have controlled that one by using potassium acetate, and this boronate ester reacted with this chloropyrimidine in presence of palladium catalyst to get the final product. They purified this product by dissolving this crude material of the product into IPA and water with the ratio of like 2.5 is to 1.2, followed by evaporation of the solvent and again redissolving the material in isopropanol and cooling that reaction mixture up to 5 degrees Celsius, and they got the pure product. That is the overall reaction process. Yeah, thank you.